Good day. I am Dr. Lyndon Waterman of Bayview Lab. I'm the laboratory manager here, and I'd like to say a little bit about Bayview Labs today. Baview Laboratory was started in 2021. During the height of the COVID pandemic, we saw a need to increase the testing services available in Barbados, and Baview Lab was formed in order to complement the range of tests that were available. Since then, we've offered testing to thousands of clients, both local and international, people who are traveling overseas. And we have also expanded our range of services to include testing for human papillomavirus, HPV, for sexually transmitted infections such as chlamydia and gonorrhea, and also for tropical fever viruses such as Zika and, of course, dengue. Today, we will be showing you the processes that we perform at Baview Laboratory, starting with reception of the samples by our trained technologists, labeling the samples to ensure your anonymity, storing the samples when necessary, and also performing the analyses on the samples. The next stage of the process is extraction. The goal of this stage is to release the nucleic acids from the human cells, bacterial cells or viral particles, and it is the nucleic acid that we will be testing to determine if the organism or the strain of interest is actually present in the sample. This is performed by first preparing extraction reagents and mixing these with the sample. The extraction reagents accomplish the goal of breaking open the cells and releasing the nucleic acid. At the end of this process, the nucleic acid is dissolved in a small volume of buffer, and that is carried through to the next stage. In this stage of the process, we prepare a master mix. This consists of reagents which we will mix with the sample, and when these are placed in the PCR machine, this will tell us whether the organism of interest is present in the sample or not. The master mix preparation can be done either manually or automated on our precision liquid handling instrument, the ML Prep. After preparing the master mix, this is aliquoted into the wells of a PCR plate, and this PCR plate is sealed and placed in the thermocycler. The thermocycler is then run on the appropriate program depending on the organism that we're attempting to detect. And while the results at the end of the process may appear to be complicated, our trained technologists are able to determine this accurately and say whether the organism is present or not in the sample. We hope you enjoyed this brief insight into the processes at Baview Laboratory. To recap, we offer testing for COVID, human papillomavirus, sexually transmitted infections, and tropical fever viruses. Soon, we will be offering a full range of medical laboratory testing services. Thank you for choosing Baview Lab for your medical laboratory testing needs.